So I did my project on ancestry mapping. Why ancestry mapping? I'm just going to quickly give some background information. The most popular sequencing method throughout the final quarter of the 20th century was known as Sanger sequencing. In the 1990s, what we today call next generation sequencing began to emerge. These new methods paved the way for entire genome sequencing. There were all sorts of very innovative technological developments by competing companies like Illumina. Next generation sequencing methods are high throughput, highly parallel, and maybe highly priced at one time, but those prices are plummeting really fast. So that's sort of the how. As for the why, well, one big reason why is the potential medical gain for knowing ancestry. In cases where particular populations correlate with some disease, but our knowledge of the causal mutations might be incomplete, that can be really helpful. So yeah, ancestry mapping can help predispose people to potential health risks as we move towards personalized medicine. And finally, ancestry mapping is of interest to human culture. It's the dawn of a day and age where all of us may have access to our own genomes, our own source code, if you will. Ancestry mapping may provide an outlet for some of those innately human questions that we all harbor, like where did I come from, or who did I come from? Some people may long for a sense of identity or family or uniqueness, and ancestry mapping will be of service to them. And this is evidenced by the fact that the ancestry mapping service 23andMe state these to be their goals. When individuals from divergent populations breed, let's say this happens just one time for the sake of argument, successive generations will have arbitrary stretches of genome from the other population, and that's due to recombination. Here I state the problem. Given genotype data for distinct populations, define a method for accurately determining local ancestry of a haplotype. The algorithm should also perform with high accuracy when one population is unknown. Shown below is the basic structure of my approach. I acquired genotype data from the HapMap on 165 individuals, 1,000 SNPs from each. Those SNPs are in order. Then the next thing is to calculate the major and minor allele frequencies from each population, and that is useful in the tricky part, which is to come up with an algorithm that accurately determines local ancestry, even when some segments have unknown ancestral origins. These are just the details of the HapMap data I used. So I included this slide just to sort of point out how I came up with my algorithm, because I think it sort of lends some insight into what my algorithm actually is. So in neuroscience, which is my background area, graphically representing data, especially when that data is really large or really complex, or you're really not sure how to approach analyzing the data, it's sometimes really useful to visualize it in some sort of a way, or to visualize it in many different ways and look for patterns yourself, and then tackle the problem in some way that might then become clear to you. And so that's sort of the way I jumped into solving this problem. So let's jump into how I solved this problem. But first, let's start from the first step. The first thing that I did was calculate major and minor allele frequencies. I did this, of course, using those 165 haplotypes that I gathered from the HapMap. So, of course, the large majority of the time, there's only one mutation that occurs in a particular SNP. So calculating allele frequencies really just comes down to calculating the major or the minor allele frequency, and then the other one is easy to figure out. So if you look at this chart, this chart is showing at one particular SNP what the major and minor allele frequencies are for populations 1 through 3. And the one other point I want to make on this slide is that if you suppose that some individual that was, of course, not used in calculating the allele frequency values has T, let's say, at this particular SNP, we can say that that individual is just marginally more likely to be from population 2 than population 3, or population 1 for that matter, since population 2 has the highest allele frequency for the highest allele frequency for T at that particular SNP. I place heavy emphasis on the word marginal here, but my point is that if there's a little bit of information about the test individual's local ancestry at one SNP, then there must be a lot of information potentially available if we were to look at many SNPs, say 1,000 SNPs, as I've done here. Okay, so my goal was to produce some sort of a visualization, and I figured that something useful for doing that would be a statistic that I could apply to each SNP, because then that would allow me to plot the SNPs on the x-axis and the outputs of the statistic on the y-axis. And so this is what I came up with. The way this statistic works is if you first set one population aside as a reference population, it could be any of the populations, and we're given a test individual, we want to know this individual's local ancestry, then we can look at which allele this individual has at each position. And the first thing we see is what is the frequency of that allele in the reference population. And we subtract from that the overall frequency of that allele among all of the three populations. And then what you can see is that if that value returned by the statistic is positive, then the individual or that location of the individual's genome is more likely to be from that reference population. And so this is good. We could use each of our populations as the reference once, therefore producing 3 output vectors, and we could produce 3 graphs. The question is now how to visualize this data. Shown here is the first attempt I made at visualizing these 3 output vectors, having size, each of those vectors having size 1 by number of SNPs, so 1 by 1000 in this case. And it's using heat maps. And the benchmark here is going to be accuracy. By the way, I think I mentioned that on the next slide. And the way a heat map works is by mapping a spectrum of colors to a set of values. As you can see here, negative values are indicated by cooler colors and positive values by warmer colors. Warmer colors are going to suggest that the local region of the genome spanned by those SNPs derives from that population. The problem with this method is that there's no good way of systematically determining where the switch points are or the locations of recombination are. This would have to be done manually. One idea might be to have a threshold value, but that won't work because there's just so much noise here. So even when you're seeing warmer colors, there's more positive values, there's still going to be a lot of interspersed negative values. It's just that the trend is for warmer colors or the trend is for cooler colors. And so this was yielding about 80% accuracy. I determined a much better or more useful approach to visualizing the data, and that's to plot the elements of the output vector as some sort of a bar or stem. And I'll get to exactly why I found that to be more useful. So I included a few slides here just to make absolutely clear my interpretation of the data. So you have top, middle, and bottom, each representing a different reference population. Look for where the stretches of positive values are, and that designates ancestry. So you can then drop them down and piece together the local ancestry of the individual's genome. So based on the data I've been showing, it's pretty easy to intuit about where recombination is occurring, but the next task is to compute that. This was not straightforward because, again, the data is so noisy. What I did was fit a smoothing spline. A smoothing spline is a smooth curve obtained by filtering out noise. It involves a parameter p, the smoothing parameter. And what you can do is simply take the x-intercepts at this point to determine the locations of recombination. Here I show all 3 output vectors. The smoothing splines are overlaid in red. We can simplify this to just the important bits of information by plotting a step function with the x-intercepts. The dashed lines here show the true locations of recombination. Now we can ask the question, what if one of the populations is unknown? So, unknown. And clearly that's okay using this method because presence of an unknown population will be indicated by the lack of a positive trend in any of the known populations. So you'll see a gap. So for some parametric analysis, one thing I did was vary the smoothing parameter p. So that's shown here, top to bottom, p is getting smaller. And I settled upon a p-value that worked best. I suppose I shouldn't call it p-value though because that's confusing. The smoothing parameter. Another thing I did was vary the number of SNPs to see just how few I could get away with. I had to make some adjustments such as to the smoothing parameter, and you can see the shaded gray regions are where the red line, the smoothing spline should be positive, and for the most part it seems to be doing a fairly good job, and indeed accuracy was greater than 97% still. With just 100 SNPs, accuracy was still greater than 95%. It came as a bit of a surprise that the number of SNPs could be as few as 40 and still achieve accuracy. Just looking at the figure, you can see that the actual locations of recombination denoted by the dashed lines align fairly closely with the x-intercepts of the spline. And finally, here's a summary slide just showing my results. These particular results are based on 1,000 SNPs and 4 recombination events. Accuracy was greater than 97%, and this is much better than baseline, which was 80%. The reason that this latter method outperformed baseline was because the baseline method necessitated manually determining the locations of recombination, and there's a lot of subjectivity in that, and that's also a stage where bias and human error can come into play and affect your results. So this method is accurate. Accuracy was greater than 90% for as few as 40 SNPs, compared to baseline, which received 80% accuracy using 1,000. This method is fully automated and fast. It's faster than baseline, if for no other reason than the fact that the baseline method requires manual labor. As far as future directions go, there are many future directions for this type of research. Recombination is not entirely random. There's recombination hotspots, so it will be interesting to study how this information can be used to fine-tune analysis. Of course, cheaper sequencing will cause the store of genotypic and phenotypic data to grow ever larger, and more populations will eventually be represented. Finally, the emerging field of bioinformatics will play an important role. Programming was done in MATLAB. My name is Yancey. Thank you.